Hello everyone, I'm Benny, and today I will be talking about what is animal cloning and what it is used for. Why do I choose this topic to talk about? It is because when I was little, I once imagining about having another Benny who can play with me and have the same unique idea just like what I have after watching an episode of Doraemon. This cloning project may help myself and the audiences to discover deeper about what animal cloning is. Why do humans need cloning technology? Cloning technology can benefit humans because of its several usages. It can be used in cloning animals' models of disease and clone stem cells for repairing damaged organs. Cloning animal models of disease is a method to examine harmful diseases found in human bodies by transferring the pathogens into lab mice in order to have a deeper understanding of the effect. Lab mice are used because their DNA are 97.5% similar to ours, and ethical problems may cause if we use the human bodies directly. Cloning stem cells for repairing damaged organs is another method which the cloned stem cell can grow into any other cells and replicate to repair the damaged body. There are two major parts of animal cloning: the artificial twinning and the somatic nucleus transfer. Furthermore, the somatic cell nucleus transfer includes two other techniques: the Roslin technique and the Honolulu technique. Two of the techniques are quite similar, but one uses an electric pulse to fuse the nucleus and the egg, but the other uses chemical solution. How does the artificial twinning works? The artificial twinning is actually doing the same process as the natural twinning. The egg and the sperm will first meet and then fuse together into a fertile egg, which is also called a zygote. As the zygote develops into an embryo, scientists or doctors will make it separate into two embryos. The embryos will then be placed into the petri dishes and wait until it has grown into a certain size. After the embryos are growing into a certain size, the embryo will be moved to the surrogate mother's uterus and have the same treatment just as a normal baby. Okay, now let's talk about another cloning technique, the somatic nucleus transfer. The scientist will extract the target organism's somatic cell and have the nucleus sucked out. Then, the scientist will find another same species organism and extract her egg. The scientist will then fuse the nucleus removed egg and the somatic cell's nucleus together. This step also distinguished Roslin technique and the Honolulu technique by the way it fuses the zygote. Now I'll introduce you the most well-known example, Dolly the cloned sheep. Dolly is born on July 5th, 1996 and has the same appearance just as the Roslin, which offered the somatic cell nucleus. Many cloned animals have a neonatal problem, but not on Dolly. Why does Dolly have three moms for parents instead of a mom and a dad? Dolly has three mothers: a mom that provides the egg, a mom that provides nucleus, and a surrogate mom. How is Dolly cloned? The scientists first extract the somatic cell from the white sheep and have the nucleus sucked out. Later, the scientists find another same species black sheep and extract her egg. The scientists then fuse the nucleus removed egg and the somatic cell's nucleus together and then put it into a surrogate sheep's womb. Dolly was born in a few months later. However, Dolly has some problems on her joints, which usually only happen on the old mammals. Dolly even died in a very young age to sheep. Afterwards, the scientists found out that the zygote that developed into Dolly has its nucleus from a grown-up adult sheep. Due to the old nucleus, the telomerase of the Dolly is shorter than sheep that is similar age to Dolly. Why does the telomerase affect Dolly's lifespan? Telomerase is on the tip of each chromosome. According to the scientists' research, it is made up of 15,000 bases. Telomerase is used for protecting the rest of the bases and make sure they will be replicated because the polymerase can replicate the end of the DNA. The telomere becomes shorter during every replication of cell. As a result, Dolly's aging can due to the unusual telomere length she has. What are the problems of gene cloning technology? The distinction of a certain species. Animal cloning may decrease the variation of a species. If the cloned animal has dominated the species, a distinction will occur if the environment has changed. The second is the new disease may be discovered. Cell mutation is a very real possibility with genetic cloning. This may result in a new and more aggressive genetic disease to begin with the new human race, creating a lot of problems. Many people believe that an event like this will be the demise of civilization. The third is the most important, the ethical problem. Nowadays, the societies don't allow the human cloning because it is against the ethical. Animal cloning techniques are now even criticized by the World Animal Protection because they think it is cruel to the animals. This is the end of my video. Hope you enjoy it.